All right, so last time we talked about photosynthesis, we were looking at specifically the light reactions that were occurring in the thylakoids right here of the chloroplast. We talked about the products from that and the byproducts from that. The major products being, of course, NADPH and ATP. ATP can be used as a direct energy source, but NADPH is going to be carried forward to be used in the synthesis part of this Calvin cycle inside of the chloroplast. We also had oxygen as a byproduct. That was from the very first step of photosynthesis. It was the splitting of water. And so when we split water, we had protons, the hydrogen protons, but we also had oxygens kind of floating around as well. So the oxygens will make their way out of the cell. And so now what we're going to do in this lecture is talk about the synthesis part. We're done with the light reactions. Now we're going to be talking about the light independent reactions. All right, so where are the light independent reactions going to occur? They're going to be occurring right over here in the stoma, or sorry, the stroma. See, it's easy to get those two confused, the stoma and the stroma. The stoma, if you remember, that's the pore underneath the leaf of a plant, where the guard cells open and close so that they can open and close that pore, and that just lets gases diffuse in and out of the leaf so that you can get gases in and out of the cells and then into the chloroplast as well. So don't confuse stroma and stoma. Like I just made the mistake just a couple of seconds ago, so it's really easy to make that mistake. Just hard to remember sometimes, the stroma versus the stoma. All right, so we're going to look inside of there, and we're going to jump right into the Calvin cycle and kind of take a quick overview of what we're going to see here. The Calvin cycle is unique to autotrophs that make their own food from carbon dioxide. So we only see this in chloroplasts. We only see the Calvin cycle in autotrophs. We do need some requirements. Well, luckily for us, we got ATP and NADPH from the light dependent reactions that we just saw. And CO2 is very readily available for plant cells or anything that can do photosynthesis as long as it's exposed to the atmosphere. So we've got CO2, carbon dioxide, we've got ATP, we got NADPH, and these are all the locations that we can find it. ATP and NADPH are found already in the stroma of the chloroplast, because if you remember from the light dependent reactions, they were already just dumped right into the stroma from the thylakoid from that whole process. NADPH was the final electron acceptor, and it's still holding on to that energy source. That electron still has energy, so we're going to be using NADPH kind of as like a stored battery. And then ATP was created using ATP synthase when we pumped all those protons across. All right, NADPH will provide the electrons and hydrogens needed to build the sugar. ATP is going to be the direct energy, and the CO2 will actually be providing the carbon in the sugar. If we remember from Unit 1 when we talked about macromolecules, when we learned about carbohydrates, we learned that all carbohydrates have some type of structure where it's a 1 to 2 to 1 ratio of carbon to hydrogens to oxygens. So we do need carbons in order to make carbohydrates, and sugar is a carbohydrate. So where do we get those carbons? Those carbons come directly from carbon dioxide. All right, and if you kind of think of it in the big picture, like where are all those sugars used in plants? Well, a lot of them are used to build polysaccharides like starch and cellulose. So if you take a look at a tree and like the bark of the tree and actually the structure of the entire tree, it's kind of weird to imagine that all of that came from a gas. Like all of that tree matter came from carbon dioxide, which is kind of crazy to think about. But we'll learn in the Calvin cycle that's exactly what happens here. And this is just a quick overview of what we're going to take a look at. We're going to need 3 molecules of carbon dioxide to kind of kick this off, this whole reaction. And it's really important when we take a look at this entire cycle of like how we're taking carbon dioxide and we're making glucose from it. It's really important to pay attention to the carbon atoms themselves. Okay, so what we're going to do is we're going to break this cycle up into 3 specific phases. We got the carbon fixation phase, and we're going to be counting the carbons as we go along there. We've got a reduction phase, and that's actually when we are going to make glucose. And then we have a regeneration phase where we actually have to recreate all the starting products so that we can continue this cycle over and over again. So let's just jump right into it and talk about the first phase, which is carbon fixation. So phase 1, carbon fixation. CO2 combines with 5 carbon molecule called RUBP. So we already have this 5 carbon molecule that exists here. It's called RUBP. It's got 1, 2, 3, 4, 5 carbons. And we actually have 3 of them right here. So we have 3 RUBP. So 3 times 5 gives us a total right now of 15 carbons. Okay, so just kind of keep that in mind. Whoops. And through this enzyme that we're going to talk about right now, this is called the Rubisco. This is a very important enzyme in the Calvin cycle. It's basically the one that kind of kickstarts this whole thing. Rubisco is one of the more important enzymes in biology that takes one of these RUBP molecules and combines them with one of these carbon dioxides. And carbon dioxide has 1 carbon. So now we have a 6 carbon molecule, 1, 2, 3, 4, 5, 6. And that molecule actually gets broken up over here into something called a 3-phosphoglycerate. Okay, so this intermediate here, RUBP combined with CO2 to form this intermediate, and this intermediate gets pretty much immediately broken up into 2 3-carbon molecules. So again, counting the carbons, making sure they're all accounted for, and we have 3 carbon molecules. And how many of these do we end up generating? Well, if we have 3 RUBP from before, so that's 15, plus 3 more CO2, so now we're at 18. We should be generating 3 6-carbon molecules, still having 18. But then when we break those in half, we end up with 6 of these 3 PGAs. 3 PGA is just basically an abbreviation of 3-phosphoglycerate. All right, and that's the first step. We fixated this carbon on the CO2 onto RUBP, and we created this final step, 3 PGA. The next step is to go through a process called reduction, and now we're going to take those 3 PGA molecules and we're actually going to do something to them to kind of modify their shape a little bit, but we will still have a 3-carbon molecule at the end of this. Okay, so we're taking a 3 PGA and then we're going to be modifying it a couple of times and then ending up with G3P. G3P stands for glyceraldehyde 3-phosphate. This is basically when we are combining 3 PGA with a phosphate group. So we phosphorylated it. That's a word that we learned in Unit 2, where we phosphorylate. I think we mentioned it also at some point in this unit, where 3 PGA got phosphorylated and then turned into glyceraldehyde 3-phosphate. But where did that phosphorylation come in? Well, we know that the phosphate group that joined this molecule right over here had to come from something that has phosphate groups that are basically ready to come off, and so ATP is going to be that molecule. ATP has a spare, not really like a spare, but it does have several phosphate groups, and so when it donates one of those phosphate groups, it releases a lot of energy, and then that phosphate group ends up being attached to this molecule, so it's phosphorylated. The other thing that happened is that we had to break up that bond in NADPH. And so this is when that NADPH from the light dependent reaction comes into play. And so we use the energy from the NADPH and turn it into NADP plus when we use all of that energy to kind of make this G3P molecule. We do this reaction 3 times to make 6 G3P molecules. So like I mentioned before, we had 6 3-PGAs, and then we do that reaction. That's because we did this whole reaction 3 times. And then we end up with 6 G3Ps. We need to do this 3 times in order to do that, so that we can make 1 glucose molecule. Because if you remember, glucose is just a standard carbohydrate, it's a sugar. It's got 6 carbons. It's C6H12O6. So 6 of those carbons came from the 6 carbon dioxides. I don't know if you can kind of like picture the first part of the reaction, or I'll just go back here a slide. We had carbons here, and so we need 6 of those carbons in order to make glucose, but that'll make more sense in just a little bit. The last phase is something called regeneration, and this is where we need to remake that RUBP from before. So this RUBP does look familiar because we just saw it a couple slides ago. This is what we started with, right? And so we need to be able to make that product again. And we, again, have to burn some ATP in order to do that. 5 of those G3P molecules that we used before are recycled, and only one of them got used to make glucose. So we only used 1 of these G3Ps. Even though we made 6 of them, only 1 of them went to glucose. The other 5 went back here to get recycled and be used as turned into RUBP. And so if we count carbons again, if we have a 3-carbon molecule and we have 5 of them, that's a total of 15 carbons. We can turn those 15 carbons with some enzyme and some energy to a 5-carbon molecule, and then we end up with 3 of them. So that's part of the regeneration phase. And like I want to mention before, we only ended up with 1 G3P molecule, and those 3 carbons came from the 3 carbon dioxides that we see here in the first step. So 3 carbon dioxide ended up turning into these 3 carbons in G3P. Okay, even though we used all these carbons here in the 3 RUBP, these are actually recycled from the previous step. So this is where it's coming full circle, right? In this regeneration step, we ended up regenerating these 3 RUBP, but then we end up using them pretty much immediately in the carbon fixation step to make that 6-carbon molecule. So in reality, in this whole cycle, we're only gaining 3 extra carbons, even though there's a bunch of carbons here. There's only 3 being gained, and then those 3 are then being turned into 1 G3P molecule. Okay, how many G3P molecules do you think we need in order to make a glucose? Well, if glucose is, again, 6 carbons, and this is only a 3-carbon molecule here, we're going to need do this whole process 2 times, 2 whole circles, in order to get 1 glucose molecule. All right, so here's the end result. After completion of the carbon cycle, we end up with 1 G3P molecule, which is a 3-carbon molecule. G3P is used to make glucose, which is a 6-carbon sugar, and therefore we need to complete the Calvin cycle twice in order to get enough carbon to make 1 glucose. So that's pretty much what I just mentioned before. And here's just another summary. Calvin cycle uses NADPH, ATP, and CO2 going in, and what comes out is a 3-carbon sugar, which is G3P. So we need to do this whole thing twice in order to make sure that we get a 6-carbon sugar. Okay, we're going to move on to talk about something that is a little bit not off topic, but it's just kind of a tangent to what we're going to be talking to, and it's going to tie in very soon on the next slide. But we need to discuss basically anatomical differences between different types of multicellular photosynthetic organisms, specifically eukaryotes. So if we take a look at like differences between C3 plants and C4 plants, these are just 2 different categories of plants in which they do photosynthesis just a little bit differently, specifically the light independent reactions. The light dependent reaction is going to be the same. They both have chlorophyll. They both do that part the same. What's going to be just tiny bit different between C3 and C4 plants is how they are going to do that light independent part. And so you can actually see the differences in the structure between these C3 and C4 plants, where C3 plants have stoma on the bottom, multiple stomata open. They have this bundle of cells called a bundle sheath, and then the mesophyll cells are going to be pretty much all clustered throughout here. Whereas in C4 plants, they still have the stoma, and they have a vascular tissue on the inside, and mesophils surrounding that vascular tissue. So structurally, they look very different when you look at them on the inside. On the outside, you might not be able to recognize them right away, which one's different and which one's a C3 plant, which one's a C4 plant. There's also another variety called a CAM plant, but we'll get to that in just a couple of slides here. And we'll mention how they do things differently. But we want to start off with talking about photorespiration. And there's a specific problem that we encounter with C3 plants. C3 plants, again, are going to be these ones over here, where their stomata are pretty much open all the time. And the mesophyll cells are pretty much exposed to the stomata being open. So if CO2 were to come in and O2 were to come out, these mesophyll cells will be able to take advantage of that pretty easily. So C3 plants have a little bit of a problem with the light independent reaction, and that problem occurs when rubisco, that very first enzyme that is fixing the carbon dioxide to RUBP, instead of grabbing onto CO2, it's going to grab onto oxygen. So I'll actually go back a few slides to kind of show you that point. So imagine instead of CO2 being here, it's just oxygen. And Rubisco would not know the difference. It would just take that oxygen and fixate it to the RUBP. And that is not going to get us our 6-carbon sugar. That's basically just going to be a waste of ATP. And that's a big waste of energy. And this can happen for C3 plants. And this happens on very hot days when the plants will close their stomata to stop water loss. So when the plants close their stomata, CO2 cannot get in. O2, which is a byproduct of the light dependent reaction, will be available, and it'll start building up inside of the cells. There's a concentration that's building up if the stomata is closed. And so what results in that is that there's not going to be any sugar production in the plant. ATP is being wasted, and that's going to be bad for the plant. It's just overall not great. So here are some adaptations that we have kind of built inside of the cells. Plants that live in very hot and dry climates have evolved to have alternative mechanisms for carbon fixation. One mechanism is to close the stomata, but that is not really good. So C3 plants do that sometimes, and those are going to be plants that have a higher tendency of suffering effects if they don't have an adaptation. They will die first, and if they just keep their stomata closed. So if they're exposed to hot, dry climates more often, they'll suffer first. But other plants that live in hot, dry climates regularly or can survive drought or very hot climates, they have these different mechanisms. So the first one is a C4 plant. A C4 plant has spatial separation of steps during the light independent reactions. The stomata will partially close to conserve water, right? Because remember, the stomata down here, if it's open, we can let gases diffuse, right? CO2 can go in, oxygen can go out, but what else can go out is water vapor. And so we want to make sure that we keep that water vapor inside. So if we partially close it, like with a C4 plant, then what will happen is that CO2 that we brought in will get actually fixated into a 4-carbon molecule, like an organic acid. That 4-carbon molecule is then transferred to bundle sheath cells, and the released CO2 is used in the Calvin cycle. Lots of corn species do this, grasses, sugarcane, things like that. And so we add this little step right here, this box, into the C4 pathway. So this is the normal C3 light independent reaction. We have CO2 coming in, Rubisco, 3-PGA, the whole thing. We get sugars coming out of that. And then we end up having this extra step in C4 plants where CO2 gets converted into oxaloacetate. Oxaloacetate turns into malate. Malate is then used inside of the bundle sheath cells into pyruvate and PEP and things like that. So what's the benefit here? Why do we take this extra step? Well, we can build up a higher concentration of malate inside of this mesophyll cell. Like let's say during nighttime where the temperatures drop down, we can, if we are the C4 plant, we can bring in a bunch of carbon dioxide at nighttime and then turn it into malate. And during the daytime, when we have to keep our stomata closed, we're not bringing in as much CO2, but we've built a reserve of malate, which we can then convert back into CO2 throughout the day. So this is why sometimes the light independent reactions are called the dark reactions, especially for like C4 plants, because that will enable the plant to be able to do this part specifically more during the nighttime. Cam plants are another type of plant that will do this type of thing, but they specifically open their stomata at night and close during the day. And so the CO2 is incorporated into organic acids and stored in vacuoles. So during the day, light reactions occur like normal, and CO2 is released from the organic acids and incorporated in the Calvin cycle. A lot of things that grow in hot climates like pineapples, cacti, succulents, things like that, they are considered cam plants, and they will do this extra step. So this is one that's specifically cut into night and day. The difference in C4 plants is that it can happen like in a night and day cycle, but it doesn't necessarily have to, because C4 plants are adapted for not just hot climates, but they are adapted to resist drought. So for instance, like corn, which is a C4 plant, it can live in hot climates, but doesn't mean that it's adapted to it just like a cam plant is. So these reactions in the mesophyll cell, they can occur during the night, but they can also occur during the day. But in CAM plants, this is specifically going to be cut into night and day. Okay, so here's just a last bit overview between the different types of light independent reactions. There's a great video that you can watch down here to kind of cover basically everything that we've talked about in the past couple slides. I will put the link to that video in the description below if you are watching this on YouTube. All right, so that basically concludes all of photosynthesis, the light dependent and independent reactions. What I would like for you to do now is complete this project in Canvas. This is called a foldable project. All the details can be found there, but it's basically a really good graphic organizer that will help you to understand and organize all of the steps of photosynthesis from the outside of the chloroplast to the light independent reactions in the Calvin cycle.